Alright, phospholipid bilayers. That's the next one. How it's insane in the cell membrane. Hey. So let's recap the differences between animal cells and plant cells, because this is crucial on every single test and no one wants to flop these tests. What do animal cells have that plant cells don't have? Easy. Animal cells have a cell membrane only, whereas a plant cell has this big boy cell wall. See, it has a cell membrane and a cell wall that's rigid on the outside because we have a skeleton, so we don't need a cell wall, whereas plant cells actually need a cell wall to support themselves, structural support. They have many small vacuoles for the storage of food and other necessary nutrients, whereas the plant cell has one large vacuole. Once again, the one large big boy. They have Animal cells have lysosomes and they also have centrioles, whereas the plant cells have chloroplasts with the green pigment chlorophyll, so it can generate energy from light. Now let's talk about the chemical compositions. By the way, this is definitely going to be a two-part video, which is why I'm going so fast because this presentation is huge. Kind of like my anywho, major elements in plant and animal cells. There has to be carbon, hydrogen, oxygen, and nitrogen in every single cell. They're organized into four major groups of organic compounds: the carbohydrates, which are the sugars, the starches, and the cellulose. There's also the amino acids and the proteins, which are your muscles, nucleic acids, DNA, RNA, etc., and your lipids, which are your fats, like the hair and the oils. So we have the four main groups of organic compounds: carbs, amino acids, nucleic acids, and lipids. We also have the four major elements of human cellular function: carbon, hydrogen, oxygen, and nitrogen. Wow, we also have water, of course. Water is a solvent. It provides the environment for all the biological reactions because it dissolves everything. That's what it does. Quick time, it just enters the cell, dissolves any nutrients and breaks them down so that they can be used by the cell. And it breaks them down into the trace elements. So into magnesium, manganese, which is one of these two, zinc, and iron. Imagine trying to dissolve cooking oil, a lipid, and water. Anywho, this is the important content. Cell membranes primarily composed of a double layer of phospholipids. All of the cell membranes are composed of two layers of phospholipids, also known as a phospholipid bilayer. Now, we know from previous context that a bilayer means two layers. Now let's analyze what it means. So it's actually known as the cell membrane, which is the plasma membrane, as we've been taught over the many years, but it's the real definition is that it's a phospholipid bilayer. It can be represented using the Fluid-Mozaic model. And that's the model that's best been adapted for understanding the phospholipid bilayer, and it's the one that we use today. So each phospholipid molecule in the bilayer has a hydrophobic tail, so one that fears water, and a hydrophilic head, which one that loves water. So the structure can be analyzed in a bit. So the phospholipids arrange themselves so that the tails are facing each other on the inside and the heads are on the outside. So let's see what that actually looks like. So this is the tail and the head broken down. So the molecule, as we see, is the hydrophilic head group is at the top right here, and this is the tail that fears water. And let's look at it in perspective. So the head, hydrophilic, water-loving; the tail, hydrophobic, water-hating. And this is it in real glance. So these circles on the top right here, they make up the hydrophilic head, and the tails in the middle, which point to each other, make up the hydrophobic tails. So it literally looks like two circles with tails inside for the whole bilayer, and that's known as the Fluid-Mozaic model. But if everything was locked up like that on the outside, then how do the cells exchange nutrient and information with its environment? Because we know that a cell is an open system, so it's always exchanging info and energy and matter with its environment. How is it doing so when the cell membrane and everything is closed off so tightly, not even allowing water to pass through? Here's how. Proteins are actually embedded in the membrane in several different areas with several different functions. This is what we'll analyze. There's marker proteins. They're responsible for cell-to-cell communication. So one cell talking to another cell, kind of like Facebook. They're all in their big group chat, and a marker protein is like a Facebook profile talking to another Facebook profile. Cell-to-cell communication. Transport proteins. They help transport the molecules inside and outside the cell. So when a molecule is requesting access or requesting to be your friend on Facebook, you have to confirm it. That's what the transport protein does. It confirms it as a friend and it allows it into the cell and vice versa out of the cell. Now we also have cholesterol in animal cell membranes, so that's our membranes, and it reduces the fluidity of the cell membrane. So the cell membrane is not always moving. So it provides structure to the cell membrane since we don't have a cell wall. So that's the role of the cholesterol in cell membranes, in animal cells only. So this is what it looks like, the Fluid-Mozaic model. Outside the cell, inside the cell down here, the hydrophilic head, as shown on the top, which loves water, the hydrophobic tails in the middle. This is an integral protein, means any molecule could just communicate with it and enter through here. This is a peripheral protein, kind of looks like a condom. This is the cholesterol that helps keep the shape, because as we just saw right here, we need the cholesterol in animal cells to reduce the fluidity and reduce the movement of the cell membrane. And we also have the messenger proteins, which are not in that picture. It's on my PowerPoint. Cell membrane is considered only semi-permeable. It means it's a selective filter. It's racist. It doesn't want anyone to get in and out. It chooses who gets in and out. Doesn't mean you have to be white, but that's white right there. White. Anywho, the ability of molecules to cross the cell membrane is determined by a molecule's one thing and one thing only. The size, which is how impermeable it is to large molecules. The charge, so if it's a positive or negative ion. If it's a charged molecule, it won't enter. If it's uncharged, it'll enter. Polarity of the substance, and the concentration gradient. That's the biggest one that we'll talk about. So what is a concentration gradient? Well, simply put, a concentration gradient is the difference in concentration of a substance across an area. Let's look at these pictures for examples. In this situation, the concentration of this molecule right here is not as much as the concentration there. So the molecules will actually move out and more will enter in there. Now, sometimes this will actually require energy. Particles naturally move down the concentration gradient from areas of high concentration to areas of low concentration. This is called diffusion. You guys have been learning this shit from grade 7, so you should obviously know it. It basically means when you spray like Febreze, the concentration of the chemical is highest to where you're actually spraying it, and then it spreads out over the room. So it goes from an area of high concentration to low concentration. That's what particles naturally do. They move down their concentration gradient because of the particle model of matter. Now let's talk of the different types of transport across the cell membrane. We have passive transport, which is diffusion. So passive transport requires no ATP energy during the transport as they naturally move down their concentration gradient, which is diffusion. However, the concentration gradient for each solute is independent of any other chemical species. So if you have carbon and hydrogen trying to enter a cell, the cell inside has a bunch of hydrogen, but it has no carbon. The carbon will be allowed to naturally move in and out because of the concentration gradient. However, the hydrogen molecule will not be allowed in because there's already so many hydrogen molecules present inside the cell. So it's independent. The carbon was not affected by the hydrogen. The concentration gradient for each solute is independent of any other chemical species. And it requires no energy, so it happens quick time. Simple diffusion is when a membrane is permeable to the solute, which means it allows it. Let's look at this as an example. This is the outside, that's the inside. We have this many molecules outside, this many inside. Now these molecules want to move over, and they simply can because there's more concentration of the molecules outside at the start than inside, so they naturally move down their concentration gradient and enter the cell. Now there's an equal balance, which is equilibrium, which is what every single cell wants to achieve. Now there's a second type of transport. It's not just diffusion like passive transport, but it's facilitated diffusion, which is still a type of passive transport. Here's how it works. The membrane is impermeable to solute, but its solute-specific embedded transport proteins allow for passage of the respective solids. Here's how that works. These are some specific proteins stuck outside the cell membrane and inside. They're called transport proteins, as we described earlier, and they'll allow a specific solute in and out of the cell because they are races. So in this case, we have the green on the outside, the orange on the outside, and vice versa. Let's look. Oh, the molecules were racist. They were denied by the orange ones, but the green ones, they were also denied. So what the heck? Oh, but they let one orange guy in. What a savage. And they let another orange guy out and a green guy in. Oh, okay, so the orange one lets the orange in, the green one lets the green in. Wow, that's some racist shit. So yeah, transport proteins allow for specific molecules to enter and leave the cell, and it depends on everything about the molecule. It's solute specific. That's facilitated diffusion.